Okay, hi everyone. So my name is Jamie. I'm a postdoc at the School of Earth Sciences in the University of Bristol in the UK. So I'm currently funded from an AXA Research Fund fellowship looking at ecosystems, or plankton ecosystems, and how they interact with the biological pump. What I wanted to show you today was some work I did as a postdoc at Bristol. This is an ERC funded project led by Andy Ridgewell. And this project was specifically designed to build a model of plankton ecosystems we could use to look at past climates. And since we can't be there in person, this is what I look like. And this is my situation during the pandemic of sitting at work with my dog. And so my dog joins me in most virtual meetings now. So I'm going to give you a kind of broad talk today that kind of tries to give you the perspective of developing this model and the kind of challenges and etc. that we dealt with. So to start with, I'm going to talk about the biological pump, what it is, what processes are important, and why we want to think about biological pump in the past. I'm going to turn to how we model plankton ecosystems, and this is going to be looking more at the sort of principles and concepts that underpin these models, and then how we decided on what model to use. And then let's look at a paper I had, which was the first application of a model of this type to looking at past ecosystems in past climates. So I think this is probably going to be very familiar to you. So this is the biological pump. So we have CO2 in the atmosphere exchanging with carbon in the surface ocean in the form of dissolved inorganic carbon. And we have phytoplankton that are photosynthesizing in the surface ocean, taking up that carbon and nutrients to form organic matter. So this is what we term primary production. And so most of that organic matter will be consumed by other hydrotrophic organisms and eventually will be returned back to dissolved inorganic carbon in the surface ocean. But a small fraction of that organic carbon will actually enter the ocean interior as passively sinking particles of particulate organic carbon. This is what we term export production. And that POC will continually sink down through the water column, and it's colonized by bacteria that will degrade it and respire that carbon back to dissolved inorganic carbon. And that carbon will now be in the ocean interior, and it's isolated away from the atmosphere in the surface ocean, where it can interact for a period of time, which is defined by the circulation time, which returns the DIC back to the surface. And so generally, the deeper it gets, the longer it takes for circulation to complete this cycle. So this circulation return part is much slower than the initial redistribution of DIC. And so we accumulate an amount of carbon in the ocean interior, which is about 1,700 petagrams of carbon. So in comparison to the atmosphere, that's 600 petagrams of carbon. And if we were to kill off all the plankton in the ocean, this DIC would re-equilibrate in the ocean and with the atmosphere, and it would increase CO2 by about 150 to 200 ppm. So quite significant, but that's quite an extreme scenario. What's more likely is that we think that this sequestered carbon pool can vary with changes in export production and with where the DIC is being released in the water column. So the way in which these change has progressed, our way of thinking about this has progressed in the last few decades. And so in the last few decades, we have progressively realized that these processes are determined by ecology and biology. So here's an modeled estimate of sort of the dominant plankton groups in the ocean. And if we look at remineralization, so here we have an estimate of basically the percent of the organic carbon that's exported that's reaching the deep sea and going from 0% to 30%. You can see, so this is an estimate from an inverse model, and you can see it has a very distinct spatial pattern. And this has been linked to various factors such as temperature. So the colder waters in the poles slow down bacterial metabolism and means that more organic carbon reaches the sea before it gets degraded. It's also linked to factors such as large cell sizes. So diatoms dominate the high latitudes, and they're very, very large size organisms. That means that they sink relatively fast. But this is kind of more complex, and so underneath what we have is an alternative estimate based on sediment trap data. And you can see that basically it's almost the inverse pattern that we see from this inverse model estimate. And in use to interpret that the particles sinking down in the low latitudes are potentially reaching the ocean because they're very dense. And this is associated with calcium carbonate shells. So they increase the density and increase the sinking velocity of those. Additionally, the activity of the organic matter might be much lower because, if you can still hear me, hopefully. So the reactivity of those particles is much lower because these particles are being reworked very heavily in the low latitudes. So we think that the biological pump function is strongly related to the ecosystem characteristics. But this is still a really big open research question as to how and why this might change in the future. So we're also interested in what implications this might have for past climates. And to sort of demonstrate this, I'm going to show you some metrics from the Cenozoic. So this is the last 66 million years of climate. And so to orientate you, the present is on the left-hand side of this plot, and on the right-hand side of this plot is basically when the dinosaurs went extinct. And so our modern plankton ecosystems have evolved from this extinction event through to the modern day. So this is why we kind of like to think about this period of time. So within this period of time, we've seen big changes in ocean circulation related to changes in paleogeography. So we've seen the formation of the Atlantic basin as we've gone through time, then the formation of the Antarctic circumpolar current. And this is a model estimate of where the deep water formation regions are at certain points during the Cenozoic. So the early Cenozoic is also characterized as a greenhouse climate. It was significantly warmer. So this is the benthic temperature of the ocean. So the first half was significantly warmer. We have no ice sheets on land. And then the second half of the Cenozoic is characterized by much colder icehouse conditions where we have an ice sheets on the poles, and we have glacial interglacial cycles down at this point down here. So we can also measure characteristics of the plankton, which are in sediments. And so this one paper characterized the mean size of those two different groups, so diatoms and dinoflagellates. And we see that those actually change with time. So in the early Cenozoic, these plankton were much larger. So this might have implications for the biological pump. So in of itself, it's an interesting question is what role did these environmental and biological drivers of the biological pump have in past climates and past carbon cycle changes? But it's also a really useful perspective on the modern biological pump. So we can look at past events and past changes to help us constrain what drivers of the biological pump are related to plankton ecosystems. So to do this, we need a model, and we need a model that can first resolve plankton ecosystems as something about the diversity. And we also need a model that can be run for timescales that facilitate us looking at the biological pump. So this is a minimum kind of 1,000 years. And we also need to run it for ability to run it for paleoclimate and the timescales associated with paleoclimate, which is typically much longer. So there are two approaches to modeling plankton functional diversity. The first is based on a model that I think you're probably mostly familiar with. So this is the MPZD style model that a few of you mentioned yesterday, where you have a representation of a generic phytoplankton, for example. So these models expand this box out into a range of different types of phytoplankton and zooplankton. So we can have calcifiers, nitrogen fixers, and silicifiers, for example. And these are based on what we observe in the modern ocean. So the parameters that define the various things like growth rates and uptake rates are derived from culture studies. So we culture different species in the lab. We measure how fast they grow, for example, and those values are then used in the model. And we end up with a more complex version of this D model, which takes into account different types of phytoplankton, zooplankton, and different types of nutrients and detritus. So this model is quite feasible to run because we haven't increased the number of traces that much. But a big problem if we're thinking about paleoclimate is that this is strongly constrained by modern species. We've literally gone out and measured how they grow and put that into our model. And we can't be consistent through time, and we know that they aren't. And we can't be sure that those species will have, the balance of those species will have changed through time. So it's a little bit difficult to apply this model to past climates. So an alternative approach that's used to model diversity is what's called a trait-based model. So these take a different approach. So rather than specifying actual groups that we recognize, these look at traits. So traits could be the rate of nutrient uptake or the optimum temperature. So here we've got 2 traits. This is the optimum light that you want to grow at and the optimum temperature you want to grow at. And the trait-based model randomly samples these the parameter space of these traits to produce theoretically possible plankton with different strategies. So here we've got 100 different random combinations of these 2 different traits. And then these 100 plankton are then initialized into a notion of biogeochemistry. And the model is run forward in time, and the plankton fight it out and they compete for resources. And some of them will win and some of them will not win. So here we have the biomass distribution of those 100 different plankton. And you can see that basically is an unsuccessful plankton strategy. So most of them are blue, they haven't actually grown. And we get some emerging competitive strategies in certain plankton. So this is corresponding to this colored space here. So this is a really representing diversity because the ecosystem and the species or the plankton that are successful are emerging out of some fundamental rules that we've defined, and we haven't defined what those rules necessarily should be. The downside to this is really slow to run. So you've got 100 traces here. That's really computationally problematic. And for example, so this run was initialized from observations, and the ecosystem was run for 10 years to allow it to reach a quasi-equilibrium. And so this model isn't really applicable in this state to be running for thousands of multi-thousand year timescales. So one way in which we took this approach and developed it for past climates is to use a slightly different set of traits. And so this is using size as a master trait. And so size constrains a lot of traits such as nutrient uptake. So this is a physiological constraint on plankton cells. So the smaller you are, the larger relatively your surface area to volume ratio is. So it means that the rate at which you can take up nutrients for your surface area is relatively can feed a much smaller volume. So it means that you can grow much faster than a larger plankton that has a much bigger relative volume to its surface area. So it has to take up relatively more nutrients to feed the bigger volume, but the surface area is not quite as large in ratio to the smaller plankton. So in general, smaller plankton are more competitive in a straight race to just consume nutrients. And this is a physiological constraint that doesn't matter about different species. And then size also constrains for zooplankton what phytoplankton you're going to go and eat. So you're not going to expend lots of energy going after a really big thing because you're possibly not going to manage to actually get it. And you're not going to go expending energy for something that's really small because it's not going to give you much energy back. So we can formalize these rules into mathematical functions. And these are in part based on theoretical assumptions and then confirmed by actual observations by culturing plankton. So generally cell size here is from small to large. And so smaller plankton tend to basically grow faster in this model. They're also grazed at a slightly higher rate. And then this shows this preference for grazing on size. So zooplankton will have a preference of a certain size range, and that preference will decrease rapidly as you get smaller or larger. So we can add these size traits into a model. So here we have we can initialize the model with a number of different phytoplankton sampled from the size range we see and zooplankton, and that's coupled with a model of nutrients and organic matter. And so the size of each plankton in this model would determine its uptake rate and grazing rate. And then we do the same thing. We run the model and these plankton fight it out for survival. And again, we get this emergent community based on these underlying rules. But by using size, we've reduced that the number of variables so we can have much less traces to save time. So this is the model that we used and developed. And to save even more time to look at longer timescales, we couple this model with an Earth system model of intermediate complexity. So Earth system models of intermediate complexity are a particular class of models that focus on resolving a wide range of Earth system components. So for example, deep sea sediments, biogeochemistry, and they often look at the feedbacks that those Earth system components are associated with. And so to do this, they often trade off some of the spatial resolution of GCMs, and they often simplify some of the ocean physics or atmospheric physics to enable us to run models for a much longer time period. So the model we're using here is called GEnIE, and this is kind of a very good example of an EMIC. So GEnIE is predominantly used for long-term millennial scale carbon cycling and biogeochemistry and looking at past climates. So it has a 3D ocean circulation, which is dynamic, but it's simplified, so it has a parameterized physics. It has a representation of ocean biology, so we have some uptake of carbon and we have reionization. That also includes, so that includes nutrients, but it also includes isotopes and trace metals. And we also have a representation of deep sea sediments and how that interacts with the carbon cycle. And you can see, so it's really good on that aspect, and you can see the trade-off that we have in resolution here. So this is the spatial resolution of the model, and you can see that it's very, very coarse. If you like really old gaming like Nintendo and Mario, you know, like I do, then it's kind of quite familiar to look at stuff like in this resolution. So it's a model that we, so this allows us to run the model particularly very fast whilst maintaining a lot of resolution for the biogeochemistry. And so we can run this model on a single core. It takes on average something like 24 hours to run a single run for 10,000 years. So it's a really good model for past climates and for running ensembles with lots of different experiments of testing out lots of parameterizations. So it's a really good test bed for something like looking at plankton ecosystems. And if you're interested, you could run this model on your laptop. I wouldn't recommend it because it's still quite a big effort. But you can go and look up the code, and we have a big manual that gives you examples of how to set it up and how to run it and do things like trying to get a snowball Earth, for example. And if you're interested in various different components of the model, I've just put some of the references down here. So we're going to now turn to the first application of this new plankton ecosystem model to past climates. And this is the first time that a model of this type, so this trait-based model, has been applied to past climates. So the question I'm really asking here is, what might a plankton ecosystem look like in a past warm climate? And so when I was revisiting this paper for this talk, I remembered actually that this was actually a really difficult paper to write, because research questions of this type that are quite broad and maybe quite fun are actually quite difficult to, once you run the model, to actually pull something out interesting out of it. So it's actually, yeah, it's been interesting to go back through it and remember actually how difficult this was. So because of this, we're going to take quite an exploratory approach. We're not going to try and pretend that we are predicting what ecosystems are. Understand how ecosystems behave in a very different environment. So the past climate we're going to use is the early Eocene. So this is the plot you saw before. So this is basically the warmest part of the Cenozoic. And the reason for this is possibly it's one of the most different climates than we have to the modern. In terms of temperature, we have no ice at the poles. And we also see big changes in the plankton cell sizes in the fossil record. And it also is a really nice, it's the best analog we have for a kind of the long-term future climate that we're heading towards. So basically a very high CO2 warm world. So the model set up, so we're going to have a continental configuration specific for the early Eocene. We're going to have a much higher CO2 than modern, so 834 ppm, so it's going to be much warmer. As we go back in time, the sun is actually slightly weaker, so we account for that process. We have a wind field which is prescribed from a GCM. So GENIE doesn't have its own dynamic atmosphere. So GCM has been run with this Eocene, a dynamic ocean atmosphere model, and we use that wind field to force our ocean model. And then for want of any more information, we are using a modern phosphate inventory or macronutrient inventory. And so I'm going to look at some key output relative to what we're thinking about. So I'm going to look at mean cell size because we can measure this directly from the fossil record. So it's an immediate thing we can compare. Looking at plankton biomass, we can relate that to abundance of fossils in the fossil record. And we're looking at changes in POC export because that relates to the biological pump. And we're going to take a very exploratory approach by turning the model on for the Eocene, and we're going to try to pick apart the different components of what's driving the changes we see. So just to orientate you, so we have the modern on the left and the Eocene, some Eocene characteristics on the right. So you can see that the paleogeography is very different. We have a proto-Atlantic, it's a little bit shallower, it hasn't not fully widened yet, and we don't have a proper Antarctic circumpolar current. We have this shallow sea called the Tethys Ocean over here. And we have this tropical free flow between the Atlantic and the Pacific. Temperature-wise, the surface ocean is much warmer because we have higher CO2, particularly at the poles. And the deep water formation is in different locations. And in terms of the ocean circulation, it's showing you the overturning circulation. And I don't want to dwell too much on it, but really it's just showing us that the circulation is much more vigorous. So how reliable is this model, given that it's such a low resolution model? We can compare it against GCMs. So this is the comparison of overturning circulation from HADCM3 and the Max Planck GCM. Looks very close to the HADCM model, and which is good because this is what the wind forcing is derived from. So that's quite positive. And it's slightly different from the Max Planck, but we do still have this kind of strong anti-clockwise circulation in deep ocean and southern ocean. In terms of the temperature profile, GENIE is actually very close to some of the other GCMs that we see. And we can also compare it against some paleoclimate data. So this is the carbon isotope from at different sites in the early Eocene compared against the model output. And you can see it does a reasonable job at recreating the large scale gradients in carbon isotopes. So for the modern setup, this is what the model predicts for these key metrics. And so very briefly, the cell size in the low latitude is very, very small. It's dominated by small things. They have very low biomass because there's very low nutrient fluxes to these regions, and they don't export relatively that much carbon. And that's compared to the high latitudes that have a higher mean size. So this is a greater diversity of cell sizes existing, coexisting with each other. It's associated with high biomass because it's high nutrient fluxes in the regions and higher export. So first what I'm going to do is turn the model on, but only turn on, basically change the paleogeography to see what impact a different circulation and a different geography has on this. So it's kind of annoying because we can't do a difference plot because the grids are different. We can see some simple things. So the low latitudes here basically remain the same across the modern and the Eocene runs. And where we see the big differences is in the high latitude. So we see a bigger increase in cell size, biomass, and POC export. And what we found when we compared this run to all of the runs that we did for the Eocene, we found, so here we're showing the zonal average of these metrics. And the gray line is the modern, and the other lines are the Eocene of various different components. And we found that this paleogeography was the biggest impact on the changes that we see. So simply by changing the ocean circulation, we're changing the metrics of the ecosystem that we see. And this is primarily due to basically nutrient cycling, changing the rate and the location of where nutrients are coming to the surface. So next we can add the CO2, so we can make the climate much, much warmer. And we can see the difference from in the metrics in this run to the previous run. But it's a little bit more complex because as we warm the climate, it has one impact on the physics, on the circulation, one impact on the metabolic rates of the ecosystem. So to separate this out, I ran an additional run where I've forced the ecosystem of this run to see the cooler temperatures of the first run. And then by comparing these different runs between each other, we can approximate what the drivers of circulation and the drivers of ecosystems. So what we see here then is that the circulation component of the temperature change really doesn't make a big difference to the mean size and the mean on the biomass of the plankton. And that's mostly driven by the ecosystem temperature effects. So here we're seeing the zooplankton grazing rate increasing with temperature everywhere, and that's pushing the biomass down at steady state of the total biomass of the plankton. And in regions where there's excess nutrients, so this is in the high latitudes, those plankton that are larger and slightly less competitive are given a slightly, have the ability to coexist. So we see a slightly increase in the cell size here. But this doesn't correspond a huge amount to the POC export. And instead, the POC export is largely driven by the change in the circulation. So again, coming back to the nutrient cycling and the rates and the location. So, like I said, it's quite difficult to do these experiments because you turn the model on and then you go, okay, what have I got? So we tried to pull out some key findings that are interesting for a kind of wider community. So one is that the physical environment is really significant when you're predicting ecosystems. And this is because the paleogeography and the circulation rates constrain nutrient delivery. So the fidelity of the modern circulation is crucially important if we're going to be modeling past ecosystems. That being said, that change is predominantly featured in the high latitudes and not the low latitudes. So this is because the low latitude regions are oligotrophic, they don't have high nutrient fluxes. So there's not much room for those ecosystems to really change. So where we see these big changes is in the high latitudes. And so what we might infer when we look at this change in mean size of diatoms, for example, is that this is a high latitude signal, not a low latitude signal. So this will control where we go and try and find deep sea sediments and trying to reconstruct ecosystems from paleo data. And then lastly, we can see that there's a mixture of physical and ecological signals when we're changing climates. And so we have to be careful when we're looking at cell size, phosphorus, trying to attribute this to a change in the biological pump, because certain drivers of ecology are going to be different to drivers of POC flux, which are related to circulation. So the one big caveat to all of this is that we didn't have those links between the ecosystem and the remineralization of the biological pump. And hopefully you could understand why, because we currently don't have a good understanding of what, even to a first order, what those drivers are. So it might be that this is a significant component when we're thinking about ecosystems. So what I'm currently doing in my fellowship is trying to find a way of looking at that uncertainty that these processes bring, and whether we can try and bring out some like fundamental lessons in a similar way to what I've done with this. Okay, so that's the end of the talk. Try to systems are important for predicting what's happening to the biological pump, available to us, and we can use different models for different approaches. And if we're looking at past climates, we need to adapt a certain type of model approach. And then we're starting to use these models to explore the role of past and the impact they have on biogeochemistry and the biological. And that's it.
Thank you very much, Jamie, for this very nice talk.